like this one. This is a good article. Let me see. Yeah, this is a really complex article. So, the people who wrote this article probably haven't read the one that I just showed you from whatever, 38 years ago. And this is a game-changing, half a page of medical literature that you don't even see reflected in the newer articles, even when there are official guidelines for the treatment of these problems. So, that's why not a lot of us, that's why some of us who, really go into detail looking at research, these are the kinds of things that we, stay up late looking for. I was reading that article last night, you know, right before I went to bed. But that's the type of article that makes a complete paradigm shift in our understanding and therefore clinical treatment of these B12 related diseases. Hello, everybody. Dr. Alex Vasquez here, coming to you mostly live. And what I'm going to do right now is go through this document, or excuse me, this image that I put together today. This was about 4 hours worth of work, and I consider myself to work pretty quickly, but this was still, just doing this diagram was 4 hours of work today. So we're still continuing the conversation on the pharmacology of vitamin B12, everything a doctor needs to know unless you're trying to win the Nobel Prize or some shit like that. So let's talk about kind of the next level of B12 pharmacology here. So I've already discussed the acquisition and absorption of B12. I talked about that in a different video. So let's assume that we now have some B12 in the blood. About 10 to 30% of it is bound to a protein called transcobalamin. And obviously that's the perfect name for a transport protein for cobalamin, is transcobalamin. When we call it holotranscobalamin, we're talking about the transcobalamin with the B12 bound to it. The other 90 to 70% of protein-bound B12 is not available to the cells. It's only available to the liver. So this is the form of B12 circulating in the blood that is available to the peripheral tissues, whether that's nerves or muscle or brain or whatever else. So holotranscobalamin is B12 bound to the transcobalamin protein in the blood. This has to meet up with a cellular membrane receptor called the transcobalamin receptor, CD320. So how many of you know what CD stands for? So CD is just a general kind of group, let's say, of membrane proteins, but the quiz question for the day for a bonus point is, what does CD stand for? And for those of you who have studied immunology and histology for that matter, but mostly immunology, it stands for cluster of differentiation. So again, these are just membrane-bound receptors, and this happens to be one for transcobalamin. So transcobalamin gets transported from the blood through this receptor into the cell. So now we're inside the cell, and I suppose maybe I should change the diagram a little bit, because at this point, now that we're inside the cell, the protein, well, I guess this is sufficient. It would happen right here, but I didn't diagram it. The protein bound to B12 is going to enter a lysosome, and the lysosome is going to degrade the protein, which is the transcobalamin protein. The lysosome is going to degrade that and release B12. So B12, now we're at step 1. I'll show you what step 1 is. For step 1, we've now released the B12 protein or sorry, we've released it from the protein after it's passed through the lysosome, and we've gotten rid of the protein, and now we can act on the B12 molecule itself. Also, within this setting of being within the cytoplasm, these are all just protein or gene products, otherwise known as proteins. These are all gene products, otherwise known as proteins. The B12 within the cytoplasm is going to be bound to another protein, which I discuss here at point number 7. And one of the tasks of that protein is that it kicks off the functional groups that are bound to the B12. So if it's adenosyl cobalamin, it's going to kick off the adenosyl group. If it's cyanocobalamin, it's going to get rid of the cyanide. If it's methylcobalamin, it's going to get rid of the methyl group. So that's also happening here, and I discussed that in this section here, number 7. So here we are. We've got holotranscobalamin. Holo is the B12 part, transcobalamin is the protein part. This goes into the lysosome, and we get rid of the protein part, so we've now liberated the B12 so it can be acted on by these other proteins. This occurs here at point number 7, I'll discuss in just a moment, and is happening here in real time, so to speak. So again, these are proteins. These in green are proteins, like transport proteins, that are gene products, and they're named after their genes. So CBLF, CBLJ, CBLC, CBLD, etc. Of these, the most common mutation is CBLC, and that's why I've kind of made it bold there. But again, the rest of these are kind of funky transport proteins. That's the way that I think about them. They almost act like enzymes, but they're not really enzymes. They're transport proteins. So if a patient has a gene mutation and they have a defect in this protein, then they're not going to be able to pass their B12 through this chain of events. Again, the most common defect in this pathway is Cobalamin C disease, which classically presents in infants, but can also present in what's called a late stage form in adults. In video number 2, or part number 2 of this series, I showed you a case of a 32-year-old man who presented with B12 deficiency and hydrocephalus due to CBLC disease. So again, these typically present in childhood, infancy, but sometimes not until adulthood. So let's go to stage number 2 now. So now we've got the B12. We've changed its charge a little bit. So now we're ready to do some action. Action step number 2 here is going to be more conversions. So CBLD, CBLE, and CBLG. That's going to convert our cobalamin into methylcobalamin, which is the coenzyme form required for methionine synthase. And that helps us metabolize this methionine cycle, converting methionine into homocysteine and kind of around and around. If we can't do this, then we, collectively in this case, are going to accumulate homocysteine, which is neurotoxic and vascular toxic and renal toxic and osteotoxic. So, the classic presentation of people who have this defect here, we call that homocystinemia or homocystinuria if we're measuring it in the blood or the urine respectively. But homocysteine is toxic. These patients present with seizures, brain damage, cardiovascular disease, renal failure, and osteoporosis. So how's that? That was pretty good. I'm remembering, I learned that from Alan Gaby in 1997. So that was 27 years ago? 27 years ago. Pretty good. Step number 4, also using methionine synthase, is the folate cycle. So here we go, tetrahydrofolate to methyl tetrahydrofolate. And again, we have another cycle here necessary for proper DNA function, whereas this is more of a true metabolic intermediate pathway. So we can go this way or we can go this way. If we go here, we're going to metabolize that same cobalamin with other enzymes. CBLD has two different types. One is called subtype 1 and the other one's called subtype 2. And you can see, instead of putting numbers, I just put the names here. So CBLD homocysteine and CBLD MMA, for reasons that will be obvious when I click on number 6 here. We're going to metabolize this with another enzyme called CBL, or a protein I should say, CBLB, and then CBLA. Again, any one of these can be mutated, and that's going to screw up our chain of events here. So you can tell that if we screw up any of these events in this first pathway, we're going to impair both of these branches. But we could impair only one of these. We could block this pathway, and then we would be deficient here, but we would be okay here. If we're deficient here, we're going to see an accumulation of homocysteine. And so if we test this branch, or maybe more accurately, I should say, when we test this branch of the pathway, we look at homocysteine levels. Again, because this involves folic acid, it becomes less specific. This is a more complex chain of events here. In this methionine cycle, it requires folic acid on this end, and B6 and betaine and cysteine and other things on this side. So an elevated homocysteine is less specific than this branch, which is much more simple, because we're simply using B12 in the form of adenosylcobalamin for the enzyme methylmalonyl-CoA mutase. And that's going to convert methylmalonyl-CoA into succinyl-CoA, and this is going to get used in the Krebs cycle, etc. Now, hopefully by now most of you know mitochondria do a lot more than just, spit out ATP. So part of what occurs within the mitochondria is also an anabolic pathway to create especially fatty acids, especially somewhat unusual fatty acids. So part of what gets created here in this pathway isn't simply getting rid of methylmalonic acid, which would be kind of the toxic intermediate here, but also the beneficial product of this pathway are certain fats that are found in the brain and in peripheral nerves. So we need this pathway to function well, not simply for ATP production, but also for the synthetic or anabolic function of the mitochondria, which is to produce nucleotides and fatty acids. So again, you can see, obviously, this is a relatively simpler pathway here within the mitochondria. So again, this is within the mitochondria. B12 enters the mitochondria in the form of cobalamin. It does not enter in the form of adenosylcobalamin. This change occurs within the mitochondria. Again, here we're in the cytoplasm. Here we've crossed the line into the mitochondria. The cobalamin gets converted into adenosylcobalamin and functions as a cofactor for methylmalonyl-CoA mutase, converting methylmalonyl-CoA into succinyl-CoA, again, either for the production of ultimately ATP or other non-metabolic metabolites, such as fatty acids that are used in the brain. So all of that's pretty cool. I'll show you point number 7, which is what I already said, and that is the intracellular binding of cobalamin to this long-named protein here catalyzes removal of moieties via dealkylation to remove the adenosyl group from the cobalamin. So let's say you go to a store and you buy adenosylcobalamin. Well, this protein is going to remove that adenosyl group, so you may as well have consumed hydroxycobalamin, for example. Same thing with methylcobalamin. It's going to remove that semi-active moiety in this in this pathway. So the argument that some have made is that, for example, consuming methylcobalamin therefore doesn't provide you any advantage because the methyl group is going to get stripped off before it even has a chance to enter the pathway. That's an argument that has been made, and that's a reasonable argument to make. Likewise, the argument has been made that no point in consuming adenosylcobalamin because the adenosyl group is going to get stripped off before it even gets into the mitochondria. Because it enters the B12 enters the cell in the form of cobalamin, not as adenosylcobalamin. So the argument that's made is that no point in taking adenosylcobalamin as a supplement because it's not going to work anyway. And that's all fine and dandy, and that's what most articles say. But I'm going to show you a case report here that turns at least half of that conversation on its head. I'm not talking about the methylcobalamin end, I'm talking about the adenosylcobalamin end. And I'll show you, I've got it right here. I'll show you my conclusion, and I'll show you the case report, and I think you'll be quite entertained by that. This case report actually comes from the Lancet, and as this was back, I think in the 1980s, maybe like 1986. I'll show you the citation in just a moment when we look at the next step, and then we'll look at the article. Some of you know, if you've been around the medical nutritional block for the last 30 years, like I have, the Lancet used to be a great journal. The Lancet used to be considered pretty much the best journal. They were at least 20 years ahead of any of the American journals. I mean, what do you have in the United States for medical journals? You've got New England Journal of Medicine, which is just drug propaganda. Same thing with JAMA. Same thing with Archives of Internal Medicine. And then the other two of the big five journals are BMJ and the Lancet. So BMJ and the Lancet were always way ahead of the American journals, and Lancet was certainly the best. And so we're going to look at this case report from the Lancet, and it's just brilliant. It's a one-page article that says more in 3/4 of a page than most journal articles these days can say, even in 18 pages. So let's go on now, and I'll show you point number 8 here, which is going to change our interpretation of this diagram. So